All right, everyone, welcome to our practical zoology session. Today, we're going to get right down to it and look at how you can tell the difference between species in the Titania genus. Ready? Let's get started.
And just so you know, everything we're covering today comes straight from this fantastic guide from your very own Department of Zoology and Entomology at Minia University. So this is designed to help you ace your practicals.
Okay, so here's our game plan. First, we'll talk classification. Then we'll compare the scolex and the proglottids. After that, we'll peek inside a mature proglottid and wrap it all up with a quick summary of the key identifiers. Pretty straightforward.
All right, first up, let's talk classification. Before we can get into the nitty-gritty of telling species apart, we've got to know where these parasites even fit in the grand scheme of the animal kingdom.
Okay, so let's walk through this. It all starts with the phylum, that's Platyhelminthes. You'll know these as the flatworms. From there, we go down to the class, Cestoda. These are the tapeworms. Then we have the order, Merazoa, which brings us finally to the genus we're focusing on, Taenia. Got it?
And within that genus, we're going to be zooming in on two really important species for our practical. Taenia saginata, you might know it as the beef tapeworm, and Taenia solium, the pork tapeworm.
Okay, let's move on to our next section, comparing the scolex. Now listen up, because this is probably the single most important feature you'll use to identify these worms. It's a dead giveaway.
So, what in the world is a scolex? Well, it's easy to think of it as a head, but that's not quite right. It's really more like a grappling hook. Its whole job is to attach, to anchor the worm right into the host's intestine. And it does this using suckers and sometimes even hooks.
Okay, take a look at this diagram. On the left, you've got T. solium, and on the right, T. saginata. Now, don't worry about the labels for a second. Just focus your eyes on the drawings right at the top of the image. Just look at the two scoles. Can you spot the big difference right away?
This is it, right here. Taenia saginata has what we call an unarmed scolex. See? It just has those four suckers. Simple. But Taenia solium, well, it's armed. It has the four suckers, sure, but it also has this structure called a rostellum. And on that rostellum is a nasty double row of hooks. Big difference.
So let's make this crystal clear. If you see hooks, it's Taenia solium, period. The key words you need to burn into your brain are armed scolex, rostellum, and hooks. That's your number one clue every single time.
All right, so we've nailed the scolex. Now let's move a little further down the worm's body and look at another really important part for identification, the proglottids. The segments. And we're not just looking at any old segment. We are specifically interested in the gravid proglottid. Now, gravid just means it's full of eggs. This is a mature segment, usually at the very end of the worm, and it's basically a little egg-filled packet that's about to break off and continue the life cycle.
So let's go back to that diagram again. Look below the scolex at those long rectangular segments. These are the gravid proglottids. And the second big clue for telling these two species apart is right there in the middle. We need to look at the uterus, the part that's holding all the eggs.
And here's the difference, laid out plain and simple. For Taenia saginata, the uterus is highly branched. It looks almost like a tree with lots and lots of thin branches coming off the main trunk. But for Taenia solium, it's less branched. See how it has fewer, chunkier side branches? This is another excellent way to tell them apart.
Okay, so now we know how to tell the gravid proglottids apart, but what about a mature proglottid before it gets packed with eggs? Let's take a look inside one to see the internal parts that both species share.
Right, back to the diagram one more time. I want you to focus on that middle drawing, the one of the mature proglottid. We're going to go through the key reproductive organs you'll need to recognize. First up, right down the middle, you have the uterus. Now, in a mature segment like this, it just looks like a simple tube. It's not branched yet because it's not full of eggs. This is the structure that will eventually grow and form all those branches we just talked about.
Okay, now look closer. See all those tiny little dots scattered everywhere? Those are the testes. And yeah, it's a bit strange, but remember, these worms are hermaphrodites, meaning they have both male and female parts. So these testes are what produce the sperm.
And last but not least, down at the posterior end, the bottom of the segment, you'll see this structure that looks like it has two lobes. That's your ovary. And as you can probably guess, this is where the eggs are produced.
Okay, we've covered a lot, so let's bring it all together in our final section. A quick summary. This is basically your cheat sheet for the lab. So, let's just run through it one more time. For Taenia saginata, what are we looking for? An unarmed scolex, no hooks, no rostellum, and a highly branched uterus in the gravid proglottid. Easy. And for Taenia solium, it's the opposite. You're looking for that armed scolex with the rostellum and hooks and the less branched uterus. These are the absolute key differences you need to know.
Okay, I'm going to leave you with one final question to think about. Something that connects this lab work to the real world. Based on everything we've talked about, especially that scolex, why do you think it is absolutely essential for a doctor to find the scolex to be 100% sure a patient is cured of a tapeworm? Mull that over. Great work today, and I'll see you in the next practical.